Aging in the wild, what beavers, mole rats, clams, and other exceptionally long-lived species can teach human beings about living longer, healthier lives. This is a webinar uh, sponsored by the Nathan Schock Centers of Excellence in the Basic Biology of Aging and the American Federation for Aging Research. Today, we are so pleased to have Dr. Stephen Austad as our moderator. As you can see on the slide, Dr. Austad is AFAR's scientific director and the principal investigator of the Nathan Schock Center's Coordinating Center, as well as a co-director of the University of Alabama at Birmingham Nathan Schock Center. When he has, in his spare time, I guess, he is also the distinguished professor and protective life endowed chair in healthy aging research at the University of Alabama at Birmingham. And perhaps most relevant for today's webinar, he is the author of Methuselah's Zoo, What Nature Can Teach Us About Living Healthier, Longer Lives, really one of the seminal works on comparative biology and aging. So without any further ado, I will turn it over to Dr. Austad.
Well, thanks, John. Thanks for the nice introduction. Let me just point out that the Nathan Schock Centers are a nationwide network that leads basic research into the biology of aging. We provide expert resources, training, and funding for researchers, and overall, we help build scientific capacity and promote collaboration to accelerate discoveries about the fundamental processes of aging, which is something that I suspect that our visitors are all interested in. There are currently 8 Nathan Schock Centers across the country. One of the directors of one of the other Schock Centers our guest today. A little bit about AFAR, in addition to the Schock Centers themselves. The mission of AFAR, which has been around for 45 years, is to advance and support healthy aging through biomedical research. And for the 45 years that we've been in existence, AFAR has been a talent incubator. We've actually provided more than $225 million in grants to more than 4,500, mostly early career investigators. And since 2017, AFAR has been the coordinating center for the Nathan Schock Centers. That is to try to get those 8 centers of excellence to actually work together to the benefit of everyone. And joining me today are Vera Gorbunova, who is a co-director of the Upstate New York Nathan Schock Center, one of the new Nathan Schock Centers. She's the Doris Jones Cherry Professor and Professor of Biology at the University of Rochester, a longtime contributor to many aspects of aging. Our other panelist is Andrei Seluanov, who's also a co-director of that Nathan Schock Center and also leader of the Live Animal Research Core of that. He is a Dean's Professor and Professor of Biology also at the University of Rochester. I have to point out that Vera and Andrei are husband and wife, and I'm always so impressed that husband and wife can work together in the same laboratory for years and years, and the marriage survives it. Congratulations to you two for that as well. So thanks for joining me. This is going to be great. This is a conversation that I really look forward to. And the three of us have a long history of being interested in the comparative biology of aging. And just for the listeners, that comparative biology is just a way of doing biology where you use a comparison among species usually to try to understand biological phenomenon. So for the comparative biology of aging, we look at lots of different species and the different ways they age, the lengths of time they live. And Vera and Andrei come at this from a slightly different perspective than I do. And I think that's going to make this a really rich conversation. So my background is in evolution and ecology. And I got interested in this whole project at first to try to understand the global pictures of aging. Why do bigger species live longer than smaller species? Why do birds live longer than mammals? A series of questions like that were really evolutionary and not mechanistic. But you two, coming from backgrounds in molecular biology, have been interested in this from what it can teach us about mechanisms of aging and how those mechanisms of aging could potentially be translated into keeping people healthy longer and longer and longer. So, with that as a background, let me just start off by asking, let's start with you, Andrei. You guys have been really focusing on long life species, species of exceptional life. Why should we do that? Most of the researchers in the field, they study mice and worms and flies, which are all short lived. What's the advantage of studying long life species?
Yeah, no, that's a good question, and I must tell you, we start working with human cells, mice as well, but then we realize that in nature we have such a big diversity in the lifespan. Like if you look at a mice, which lives only 2 to 3 years, and bowhead whale live to 211 years. Even in mammals, like you are getting almost 100-fold difference in the lifespan. But now, if you include the Greenland sharks, which live up to 500 years, or the clams, then the limits becomes almost like lifespan is almost like unlimited. What I really believe that probably evolution, in my mind at least, has no limits, no restrictions. If there's a condition when the species can survive for a longer lifespan, this will evolve to 200, 500, or maybe immortal species. But that's all speculation.
Yeah, so Vera, you guys have studied mice, right? You guys have studied short-lived animals. And when I have to say, when I came into the field and I saw that almost everybody was studying at most maybe two species of mammals, maybe mice, maybe rats, it seemed to me like a terrible failure of imagination because those species are really unsuccessful at aging, right? They fall apart, they get cancer. And I know you guys have really focused on the cancer aging relationship. Is there some reason that you think that cancer aging relationship is particularly important?
Well, yes, yes, and maybe also to add to Andrei's answer to the previous question, like why why study those? I think evolution had such a long time to solve the problem of longevity and also healthy longevity, which goes, let's say, not getting cancer. We we don't have millions of years to develop our projects in the lab, but evolution had millions of years to solve this. So it just seemed to us, well, why don't we try to understand it through the lens of evolution, how to solve the problem of longevity and disease resistance? And then the solutions are probably very good ones because they withstand the test of time and also the test of environment and adversity, and these species still live. So, back to the cancer question, I think there is really a link between cancer and aging in many ways. Sometimes there are some opposing forces of selection, but sometimes it kind of is works in the same direction because to live for a long time, you also have to stay cancer free. And there is a very strong selection for animals not to develop cancer while they're still reproductive age, because if a young animal develops cancer, it's not going to leave any progeny. That's quite certain. So I think that here the selective forces were acting in the same direction. Now, something else I can add that often when we came into this problem thinking about aging, and we want to understand why this animal lives as long. Aging is very challenging because the phenotypes are often very subtle. We have to really find what corresponds to aging, while in cancer, it is much more defined. So sometimes we started with aging question, but we ended up characterizing cancer related and saying why this animal doesn't develop cancer. So I would say it's sometimes an easier question to answer.
No, I mean, think there's also the aspect that, the war on cancer, the so-called war on cancer has been going on for 50 years now, but a lot of the tools that were developed by that actually turn out to be useful for studying aging as well. And you guys have really taken advantage of those tools. Now, one of the interesting things, the title of this webinar is Aging in the Wild. But you make a really good point. So we can knock out a gene in a mouse and it can live a bit longer in the laboratory. But that's a very artificial situation, and we don't know how well that animal is adapted to that change. In fact, if I had to guess, and I know that I heard Andrei just a few days ago say, yeah, if we took one of our long-lived animals and we put it out in nature, it wouldn't live very long. And there's no question that that is absolutely true. But one of the things we're interested in is health span, right? And health span, for animals in the wild, you don't have to worry about health span. If they're alive, they're healthy. If once they don't get healthy anymore, they're gone. So that's another big advantage, really, of focusing on these. And one of the things I hope that we get into a little bit is maybe the differences between conditions in the wild and conditions in nature. But one of the things I got interested in, I got interested in opossums, right? Opossums, because they age so poorly. They didn't live any longer than a mouse. So this whole idea that the long life species could teach us something occurred to me probably 15 years after I got into it. But the way I think about it now is if we can make something that's really unsuccessful at aging a little bit less unsuccessful, what are the chances is that's going to help humans who are already, we're really successful at aging. But you take these species that do things better than we do, that resist cancer better than we do. If we can figure out how they can do it, and you guys have made a lot of progress on that, then I think this is something that likely is going to really contribute to extending human health. Now, you guys have answered one question that I had initially already. Your work in some has done. So here was my evolutionary question that I was thinking about. How do animals evolve long life? Are there many routes to long life, or is this a big convergent evolution question so that they all end up doing it the same way? You guys have answered that question. Do you want to tell us about what you found? Vera, why don't you start on this?
Yes, that is a very big question. Is there one way? And the answer is it's probably a combination of both. If we focus on individual long-lived species, so we can find some species-specific strategies. For example, by studying naked mole rat, we found that they have a very high levels of hyaluronic acid, which people may be familiar with from cosmetics. It's sometimes part of different skincare products, but it's also normal part of the tissue that we have too, but naked mole rats have 10 times more. So we came up with this finding, and it does a lot of good things for naked mole rats, protects them from cancer, reduces inflammation. But we didn't find it in humans, we didn't find it in the whales. So that is something more unique for that sort of animal. But we also find adaptations or mechanisms that are shared among many long-lived species. And among those mechanisms is genome stability, ways to maintain DNA, minimize damage to DNA and minimize mutations. So that seems to be really conserved. Interestingly, the exact proteins, like exactly how animals achieve that, may differ. So in some species, they may enhance one protein, one enzyme, and in another group, that would be a different enzyme. But then we can kind of see that it all converges to better genome maintenance.
Right, and so let's jump into whales. I mean, you guys had a recent paper, a high-profile paper, talking about what make whales live so long. I suspect, maybe I'm wrong about this, but I suspect that you do not have a colony of whales in your lab.
We keep them in Lake Ontario.
So this brings up a question, particularly for things that live a lot longer than we do. How do you study? How do you study these things? Andrei, can you tell us a little about that?
Yeah, that's one of the challenges of comparative biology, but it's relatively easy to resolve that challenge because what we are using is the primary cell lines or primary lines isolated from this species, and we have primary cells, fibroblasts from whales, from bowhead whales, from the other animals as well. And we are collecting tissues from the other organs of the other species. Actually, right now we have this collection of over 50 mammalian species, different organs, primary cells isolated. And then we can grow these cells, we can treat them, we can see how they react to the different oncogenes, for example. Are they easy to transform or not? How the transcriptome looks like, how they respond to the DNA damage. Are they good at DNA repair or better or worse at DNA repair? This is hundreds of different or thousand different parameters can be done just measuring the tissue culture cells. And the ultimate goal of comparative biology, of course, people ask like, are you going to study the naked mole rat? That means you are going to study them for 40 years. Like that is a human lifespan almost. How you can do the experiments length of 40 years? It's not the purpose of the comparative biology. Our idea is to find the genes or the pathways on these species which allow them to live longer and then transfer it to mice. And mice lives only 3-4 years, maximum of 3 years primarily. And that's already feasible. During the PhD, we can do these experiments and we'll see the effect of the specific gene manipulation or the pathway which allow the mice to live longer. And of course, I would like just to stress that if we are working on mammals, we all have the approximately same number of genes. It means the difference between us and whales, us and naked mole rats, is somewhat different regulation of the gene expression, enzyme specificity, or activity of specific enzymes. And this is all potential drug target. It means even though we start working with this boutique organisms, you think, oh, they are so weird, they have no application to human being. But because we have exactly the same genome structure, we need just to find a way how to tweak our genes in order to behave the same way as they achieve the results in whales or the other creatures as well.
Maybe I would like to also add to it that we don't need to have a very large numbers of animals, of wild animals. So it's enough for us to have a biopsy and derive primary cells with it or do some omics research, because then we use it as a discovery platform. We find what pathway is really working differently in long-lived animals, and then we can transfer everything in cell culture, in some laboratory organisms like mice or flies, and that's why we can benefit from both worlds. We get a lot of insight from wild animals, but then we can also apply genetic tools.
Yeah, so the bowhead whale is a particular interest to me from an ecological perspective, and I think this also figured into your discovery there. It's the only one of the 10 or so species of large whales that lives its entire life in the Arctic. It lives its entire, all the others, they migrate to the tropics to reproduce and then they go back, but it lives its entire life in cold water. It's the only whale that does this. Now, for the Greenland shark, which is also an Arctic species, this makes a lot of sense that would lead to long life because the cold water would slow down everything. And the Greenland shark, doesn't swim any faster than you and I walk, right? So it really, really leads a slow life. But whales are endothermic. They have to keep a high body temperature, and cold water, that should increase their energy rate. So you would expect that might make them short-lived, but in fact, they're long-lived. So that makes them doubly interesting to me. And then, I don't know if I've told you this, but I think there's a serious underestimate of how long they live. And I say this because of the way that they do it, which is looking at this change from left-handed to right-handed amino acids in the islands. That depends on temperature. And they've assumed that the body temperature is the temperature that it's exposed to, but that islands is just a few centimeters from really, really cold water their whole life. And I recalculated the longevity using the same formula. And if I just used a few degrees lower body temperature than their core, I came up with 250 years maximum. And I think that's still probably an underestimate. So this species really has a lot to tell us. And the cold exposure plays a role in what you found, correct? You want to tell us about that? Yeah, Vera, go ahead. You could. This was one for you.
All right, thanks, Steve. Yes, indeed. I think it is an underestimate. We're just trying to rely on cases where we can well document it, but No, I'm pretty certain they live even longer than 200 years. Yeah, they are quite active, unlike Greenland sharks that just swim very slowly through life. Uh, Whales, yeah, they're active just like, just like us. So what we found was very unexpected. We really went in expecting to find, an anti-cancer mechanism. We thought maybe a tumor suppressor like P53, like what they found in elephants. There are more copies of this tumor suppressor, and what it does when cells are about to become cancerous, it just kills these cells, induces cell death. And in elephants, there are more copies, so they're killing more cells. We thought we would find something in the whale, maybe a new gene that's responsible for that, but we didn't find it. And what we found instead was that whales just don't let things deteriorate to the point when, you have to start killing cells. They maintain their genome much better. Their mutation rate was lower. They would repair if we took cells and induced DNA breaks, so they would repair it much more accurately without inducing mutations. So then we started looking for the cause and mechanism, and here we came upon the protein that we didn't even think much about, a protein that's induced by cold, and the name of it is cold-induced RNA binding protein. Um, we also have this protein, and it can be induced by cold, but the level we found in the whale is 100 times more than in humans. So even with cold, we cannot quite get to that level as the whale. And what we showed that this protein alone can facilitate DNA repair. Even if we put it in human cells, it would improve our DNA repair and make it more accurate. So amazingly, whales evolved this protein to adapt to the cold environment, but then it was also co-opted to help with DNA repair. So evolution doesn't just, pick one thing. There are always multiple avenues how things can be utilized. And that is pretty unique for the bowhead whale because uh, Steve previously asked if there are unique mechanisms or conserved. So we find it in the bowhead whale and other baleen whales, maybe to somewhat lesser extent, but we don't find this adaptation in dolphins, for example. Um, but it results in more efficient DNA repair. And now if you think about how would it benefit humans, well, we have a fairly good DNA repair, but we achieve it through different means, not by upregulating this cold-induced protein. Now, if we can find how to upregulate our cold-induced protein, that would be something new. That would actually improve our DNA repair because our evolution didn't take that route. So we can add something useful that we don't have.
Yeah.
I just would like to jump in because this really the opportunity to link it back to the original Steve question, like how we can improve human longevity based on the other species. Because, for example, we never live in Arctic water, in the cold water. We never live underground, like subterranean, like naked mole rat. We never fly like bats. And by living in these environments, these species, they evolve unique adaptation, which allow them to live longer, like CRBP protein in whales, hyaluronic acid in naked mole rat, or in bats, unique adaptation for the immune system. But they all could be transferred to human because, as already Steve mentioned before, we are quite long-lived. Maximum lifespan for human 120. Like it's very difficult to beat it. Only like bowhead whale can do it, and the other like Greenland sharks mainly. But if you'll borrow this unique mechanism from the other species, maybe we can extend even this long lifespan what we already have right now.
Yeah, if I were a betting man, I've already bet $1 billion, so I don't have any money left over to bet on this, but I would bet that it's going to be the discoveries in these long-lived species that are going to lead to the first human serious longevity extensions. So we've talked a couple of times. We've mentioned naked mole rats, and I don't know if the listeners. By the way, for those listening, if you have questions, put them in the Q&A. I know you don't, you don't see any chat, but if you put them in the Q&A, the second half of this conversation, we're going to be devoted to answering questions that you ask. So please do that. So let's talk a little about the naked mole rat. So the naked mole rat does not live hundreds of years. You know, it lives more like 40 years. What makes it so unique that we would really want to invest a lot of research into it, which we're doing, right? Andrei, you want to take that one?
Yeah, no, that's a good point because naked mole rat is lived to like 41 year maximum lifespan, probably, maybe a little bit longer. We don't have the data about this, but this is they belongs to Muridae family. It means they're very close relatives to mice, and by size, by body mass, they're also very similar to mice. It means if you compare naked mole rat, it's not appropriate to compare it to human because we are much larger and completely different species. But most appropriate control for naked mole rat will be mice, which lives only 3 years, which tells us that naked mole rat, the same size species, is able to live 10 times longer than the mice. And it also belongs to the same family. And it's really what we are studying because these species, they evolve multiple mechanisms to allow them to live longer. It's quite interesting example because evolution never works on a single gene. And maybe this is the blessing and the curse of comparative biology. Because evolution is working on entire genome. It's working on many different pathways at once. And it means what we found in naked mole rat, we see many different genes gets optimized for a longer lifespan, better protein synthesis, better protein stability, stem cells turnover, many different parameters. Almost every parameter which is linked to longevity is improved in naked mole rat. That's why it's make it so unique and important for study. In addition, we can keep it in the lab. We have the colony of about 300 animals in this building. And we can study them. They breathe quite well. They just requires the high temperature, 32 degrees Celsius in the room.
Yeah, just a small correction that they are not Murida, but they are kind of a little closer to guinea pigs. But still, guinea pigs don't live 40 years and they're much bigger. So I think that is really the differential between the size, because usually large things live for a long time. Whales, yes, elephants. But here we have some species that are really exception. And naked mole rat is one of them. They are so small, yet they live almost half of human lifespan.
Yeah, thanks for
Yeah,
correction. Yeah, they're close to Muridas, not Muridas.
yeah, they're all rodents, though,
Yeah,
right?
they're rodents.
And yeah, so one of the things that interests me about comparative biology, in order to do it, we have to have accurate information about how long species live. Now, some of the species we've been talking about, whales, whether they live 200 years, 250 years, 300, doesn't really matter. We know they're enormously long-lived, right? And same for the naked mole rat, whether it lives 35 years, 40, 45 years, it doesn't matter because it's so much longer life. But once we start working with clusters of species, then it becomes very important to know that species A lives longer than species B and species B longer than species C. And I spent a lot of time trying to validate these ages because those of us in the field know that extreme ages, and when we say a whale lives 200 plus years and a naked mole rat lives 41 years, we're talking about the longest lived ones, right? Well, anytime you start talking about records, then people start exaggerating. So I've spent a lot of effort trying to find out which longevities are true and which longevities are bogus. And that turns out to be quite important, but it also means that we can look at a bunch of relatively closely related species and try to find subtle differences. And you guys have done that by looking at a whole series of, for the most part, small rodents, one or two larger rodents. You want to tell us a little bit about your comparative rodent study?
Yes, well, rodents are quite amazing because most people, when you say rodents, people think mice, rats. But rodents are much more than that. Beavers are rodents, porcupines are rodents, squirrels are rodents. Very, very different creatures. And they have very different longevities and rates of aging. Beavers can live more than 24 years. There's some anecdotal evidence, and Steve would correct me that maybe they could live to 50 years. But the recorded zoo record is 24. Squirrels can live also over 20 years. So those pesky gray squirrels you may be chasing away from your bird feeder. I mean, we need to give them a little bit more respect because they can live more than 20 years if not killed by someone. So quite amazing group. And within rodents, if you imagine the phylogeny, we have mouse-like rodents, squirrelidae, like squirrel-like, and naked mole rat-like. And in each of these groups, there would be some short-lived and long-lived species, which makes it actually quite an amazing collection to identify convergent mechanisms that evolve with longer lifespan. So what did we find? DNA repair again. So we're coming back to this idea, more efficient genome maintenance, particularly the type of DNA repair called double strand break repair, when both strands are broken. We find that mechanism is really enhanced in long-lived rodents and long-lived animals in general. We also find some links to metabolism in shorter-lived species. They're kind of more metabolic in general. We also find links to immune system. So short-lived species tend to be more inflamed. There is more inflammation going on with them, so that means it's important to suppress inflammation. When we also started looking into like what genes control those genes that are linked to short life or long life, so that was actually extremely fascinating because we found that in those genes that are linked to short life, they're controlled by circadian network of transcription factors. So these are these control genes that regulate our circadian biology, when we sleep, how we sleep. And that's fascinating. So that's maybe to say, well, it's really important to keep your sleep patterns consistent, because if you don't, then maybe you get high expression of the genes that are linked to shorter life. And that's the purpose of this control. Let's control these genes very well. Otherwise, we kind of going into the short-lived, more short-lived phenotype. And then those genes that are linked to longer life, we found that they are controlled by transcription factors that are linked to pluripotency, or they are linked to this embryonal state. So that's kind of very early in development. That's the state that maintains, this pristine way of life, or like in the stem cells. So this network controls genes linked to longer life. And maybe many of you heard about experiments on partial reprogramming, when the same pluripotency genes were transiently expressed in some model organism like mouse, and that could rejuvenate the mouse to some extent. And that was really considered one of the breakthrough, because you can now demonstrate you can just tweak some transcription factors and you can rejuvenate the mouse. But what we observed looking at how evolution answered that question, well, evolution took actually the same approach even before scientists knew about it, is to enhance the same networks in order to achieve longer lifespan.
Yeah, right. Well, this is great, and I have a number of other things I'd like us to chat about, but I think we should take some questions from the chat. And one of the questions is about telomere biology, and this seems to be one of the things that even people that have only heard about aging biology seem to have grasped a hold of, that telomere biology is somehow the key to aging. And so the question is, what do we know about telomere biology in whales and naked mole rats? Andrei?
Yeah, that's a good question, because actually that's the start of our comparative biology research. When we in 2000 we did our postdoc, we were studying replicative senescence and telomerase activity in mice and human. And I was really puzzled, like why in human we have repression of telomerase And in mice, we don't. Like that time it was explained because we live longer, that's why I replaced the telomerase. But now we know it's primarily due to the repression of the tumor formation or premalignant transformation. Because in this case, we have the limited size of the telomeres, and when the cells proliferate too much, you have telomere shortening, and the premalignant cells will stop from proliferation. It means original probably the way of repression of telomerase in like larger organisms, what we show it happens in larger body, like what we have in human, in bowhead whales, in any species which is larger than 10 kilograms, that's what we showed a long time ago, we see repression of telomerase is as anti-tumor mechanism, because when we are getting bigger, it means we have more cells. Every cell can potentially become cancerous, and that's called Pedo's paradox, which actually explained that idea of the long-lived species. They have to have like smaller body mass probably, and if you have a bigger body mass, then you have the higher cancer risk, and you need to alleviate somehow this risk by evolving a new anti-tumor mechanism. And one of them is actually repression of telomerase. It means in terms of the evolution, the telomere shortening or replicative senescence triggered by telomere shortening is really powerful anti-tumor mechanism. And we are getting as a more like a side effect of this because we have now shorter telomeres. We have the problem with proliferation of stem cells, especially bone marrow or any kind of the epithelial cells. And that leads to the replicative senescence, which triggers the age-related inflammation and aging itself. But probably the primary evolutionary purpose of the replicative senescence is anti-tumor mechanism.
Yeah, I probably should have mentioned at the very first for the listeners that telomeres are protective caps at the end of chromosomes, that every time a cell divides, it shortens, and telomerase is an enzyme that can lengthen that, because when the telomere gets too short, the cell thinks it's a DNA damage and tends to kill the cell or turn off its replication. And that was a very, very, very good explanation of that, Andrei. Another question that showed up in the chat is whether there's an association between how long it takes an organism to reach sexual maturity and how long it lives. And that is probably a question for me. And yes, there's a very good relationship between that. And one of the most dramatic examples of that, one that it's so hard, it's difficult for me to believe, but the Greenland shark, which has this exceptional longevity, 400 years or so, they don't reach sexual maturity until they're about 150 years old. That is a very long prepubescence, I have to say. It must be very, very painful. The bowhead whale is more like 20 or 30, so it's about like a little longer than we are. But yeah, there's a good relationship between that. So next question, does it seem reasonably to investigate the extent necessary to activate that ice protein, the CI, in humans? In other words, could you just activate it by taking icy baths or something like that? I mean, people do that, right? I don't know if you guys jump in cold water, but a lot of your countrymen do. I know that.
Yeah, in many Nordic cultures that is popular, cold swim makes people feel good. Maybe when you get out of the cold water, you have all the endorphins rush. But maybe it also improves your DNA repair. So I think this is a very important question. We know that if we take human cultured cells and leave them instead of 37 degrees Celsius at 25, like room temperature, we see induction of CRBP. How does it translate to whole organism cold exposure if ice bath? I would not necessarily recommend really long hypothermia. It can be dangerous. Really, you really don't want to lower your core body temperature. But in cold swim, people, at least the surface body temperature goes down quite significantly. So we are now talking to clinicians, and we want to really get in touch with a group that studies people who practice cold swim, And this way we can maybe take a blood sample and see whether what type of exposure is sufficient to induce CRBP protein.
Yeah, I would like just to add that, when you are talking about cold exposure or cold induced protein, it's important to remember it have to be short exposure, that we are talking about some kind of stress. Like cold stress is important, is good. But when you take a very long exposure, then really have the adaptation of the body. That's a different reaction. It means the short,
Yeah, we are
is
warm-bodied organisms, so we cannot really change our body temperature, unlike some invertebrates. Yeah, if you put them in cold, then they live longer. So for us, we cannot really apply that, but yeah, brief intermittent exposure potentially can induce it, but specifically what type of exposure we don't know yet, and that's something we really want to investigate using volunteers who are already engaging in cold showers or cold swims.
Yeah, yeah, I always had the feeling that people live longer that did this because it weeded out the people that weren't, if I jumped into cold water like that, my heart would probably stop and then I'd be out of that gene pool. Um, Here's a question about cell cycle, and this is something that you guys know, that how fast do the cells cycle in whales? I mean, we know that larger animals have slower metabolic rates per cell. How does the cell cycle timing differ among these different species?
yeah,
Or is that something that you can study in the lab?
Yeah, that's a good question because actually we'll see that correlation between rate of proliferation or the time of the cell cycle and the lifespan. Actually, long-lived species, they have the slow proliferation, and whale cells, they're quite slow dividing. I know, I think they requires few days for cell division. Human cells, it takes usually 16, 18 hours for one cell division. But it's a little bit difficult to do this kind of comparison because cell division really depends on cell culture condition. And here there's a problem when we have the good cell culture condition for human or mouse cells, and we know how to grow these cells. When we are growing whale cells or capybara cells or naked mole rat cells, we always have this concern that is it the optimal condition for this unique species? Maybe we are missing something. It means in general, yeah, we see this pretty good correlation between the rate of proliferation or length of the cell cycle and longevity, but it's difficult to make this strong argument about this.
Not always. Whale cells are slower, but for example, beaver cells are they grow very robustly. So I think it varies. It may be like there are some species where there are some mechanism that kind of are sensitive and they would slow cell cycle, but then there are other species that feel very good in the culture conditions that we use, which are actually developed for human cells, and they proliferate just very well.
Thanks. So here's a question, and I swear this is not a planted question, but this is right in your wheelhouse. So could transposons be involved in accumulating DNA damage with aging? Now, let me just say for the listeners, transposons are these things that can jump around. They're these genes that can jump around in your genome. So who wants to take that?
Well, I can take it. I love transposons. Yes, transposons. That's they make the bulk of our genome, and they're called dark genome matter or like parasites. Um, so they can really influence us, like parasites living within us. Of course, they influence us. In humans, what we showed is that when we get older, transposons become more active. Actually, upon stress, they also become more active. They are like rats that trying to leave the sinking ship. You get stressed, your transposons start jumping. And that's same thing with aging. And then they induce inflammation. There is a mechanism by which transposons can trigger inflammation just like viruses, even though transposons are within our cells, they're not viruses coming from outside, but our body reacts to them as if we are infected by a virus. And that may be driving aging. Now, in terms of comparative biology, to what extent different species learned how to manage their transposons differently. We are still trying to answer that question. There is technical limitation to study transposons really well. We need to have very good quality of the genomes, which we have for human and mouse, but we don't have it for most other species. It's good enough to study genes, but transposons are not genes. They are very, very repetitive. They're very messy. And often these parts of the genome are not really getting annotated very well. But what, interesting fact is that when I was describing these mechanisms that evolved in short-lived versus long-lived, so one pathway that we noticed to be really dampened in long-lived species is induction of inflammation in response to transposons. So it seems like long-lived species actually have better way to handle transposons. We don't know if they're controlling them better or they, just don't activate inflammation as much, but there is definitely something in it, and that's something we are very interested in pursuing.
Yeah, just I would like to add in, we just received the funding from RPH actually to answer this specific question, because we are going to test inhibitors of reverse transcriptase. It's one of the enzymes from transposon for the lifespan of mice, and if it is successful, it will be go to clinic. But we'll see it in 2 years.
Then will be a clinical trial to see if suppressing transposons can extend human life and health.
Yes, this is what I was getting at when I said this is right in your wheelhouse. So here's a question, but is there a relationship between reproductive lifespan and longevity? Because women who have later menopause tend to live longer than women who have earlier menopause, and does this occur across species? I'll take that one. It turns out that something like menopause is very rare among mammals. And so usually reproductive lifespan and actual lifespan are the same in the overwhelming majority of animals. So there's a whole list of questions to answer about menopause that would require an entire other webinar, which will be really interesting because there's a lot of really interesting work going on in it right now. But for the most part, reproductive lifespan equals lifespan in most species. Another question about heart rate. Is there an inverse relationship between heart rate, maximum heart rate or resting heart rate, and longevity across or between species? Because exercise is one method in humans to lower basal heart rate, but whether this impacts longevity is not particularly clear. You guys want to take that? I mean, I'm happy to take that one
Yes, you can take it because
I've done a lot of work. I mean, there's this idea that was really probably the dominant idea in the field for a long time, that basically metabolism determines longevity, and that if you have a fast metabolism, you are bound to live a short life, and if you have a slow metabolism, you're bound to live a long life. Now, there's some fundamental truth to that is there's general trends like that, but there's so many exceptions that it doesn't make sense. One of the early researchers named Raymond Pearl came up with this idea, called it the rate of living theory, actually thought that exercise would be bad for you because it increases your energy expenditure, and even wrote a paper called Why Lazy People Live the Longest. I have to say though that Pearl died at age 61 at a massive heart attack because he lived what he said. He never exercised and he ate a lot. The folk version of this is that all mammals have about the same number of heartbeats per lifetime, and that's simply not true. There are species that have way more than other species. So that's a intuitively satisfying idea, but it just doesn't hold up in a comparative perspective. What about the relationship between animal hibernation and longevity? You guys want to take that? We know that bats, for instance, the longest living bats hibernate the longest. Do any of the species that you have been studying, any of the rodents that I know you've done some work in bats too, and maybe you just want to take this as an excuse to talk about what you know, what you've learned from studying bats.
Yeah, well, some of the species we study hibernate. I think this is kind of a big question, and people use different approaches to address how hibernation affects longevity. There seems to be a positive effect, so hibernating species seem to live longer, although I think it's harder to make a very clean test, because when animal hibernates, it means it's somewhere deep in the burrow, so predators cannot kill it. So if we study animals in the wild, that would result, of course, in seeing longer lifespan. But there were very nice studies where researchers measured, using biomarkers, methylation age of hibernating ground squirrels, and this age would actually go down after hibernating period. So that there is some uh, perhaps rejuvenating effect of hibernation, which makes it also a very fascinating adaptation to study, because like how come bear hibernates and doesn't suffer from muscle loss or some other issues, what would happen to us if we were to stay motionless for a long time, or in small rodents even their body temperature drops down during hibernation. Many hibernating animals accumulate large amount of body fat, and then like how come it doesn't mess up their metabolism? So these are all very fascinating questions that can be studied only using these non-standard animals, not your typical mice.
Yeah, I mean, this is one of the things that studying long live animals teaches, right? There's things we might learn about their longevity, but there's also other things like how do bats hibernate for 9 months and wake up and fly away when if we go to bed for a couple of weeks, we can hardly walk at the end of the time. Um, Here's a question that about metabolic rate of cells in culture. So we know that organisms of different sizes, mammals, let's say, have different basal metabolic rate. Is that kind of replicated in cell culture, or is that something that disappears when you put cells into culture? Or do we know?
We don't have the clear answer about this, especially transition from tissue to tissue culture. But what we know from tissues, where Vera talked about, that the high metabolic rate associated actually with shorter lifespan. It means when the longer you live, the lower metabolic rate you need. In general, we see very similar process in tissue culture as well, but we didn't study it too deep. And what's interesting, because again, when you're going to have the longer lifespan, it means you want to repress your metabolic rate, but you still need to have the energy to survive. And that's what Vera already mentioned before. What they are doing right now, the long-lived organisms, they're putting under control of the circadian rhythm genes, which only allow them to be expressed when we need it, like during the day when we're actually active and running. But when we are sleeping, we don't need the energy anymore, and we are going to shut it down, this huge energy production machinery, because that's how we're going to survive for a longer
But
time.
I would like to bring bats in one more time, because in general, yes, so sort of lower metabolic rate, longer life, but there are exceptions everywhere. So bats fly, and when they fly, their metabolic rate is extremely high. Their body temperature goes to like almost up to 42 degrees Celsius, and yet they can live for 30, 40 years. So they found a way to counteract or kind of compensate for high metabolic rate with some adaptations. And that is something we are studying right now. And what we are finding also genome stability, but through some other mechanisms, we're still figuring it out. Not what we found in whales. Something else that they do differently that probably allows them to maintain stable genomes despite, having this high body temperature and flying. So they do something differently.
Yeah, I just
Yeah,
would
and
like
not
to
not
add
to
that.
mention birds, not to mention
Yeah,
birds that also have the high body temperature, also have the high metabolic rate, also have exceptional longevity, right?
yeah, I have exactly the same idea about the birds, especially hummingbirds,
Yeah.
because the metabolic rate is huge, and blood sugar is like over any diabetic limit, and they just live quite okay.
Yeah,
It
these
means,
tiny birds, I think they can live to like 10, 20 years still.
yeah,
And they're not mistaken for
hummingbirds. Yeah, yeah,
hummingbirds. Quite amazing for such a really very tiny creature.
yeah.
Yeah, so here's a question about melatonin. And melatonin had a moment, right? Had a moment that it was a big, but taken at bedtime lowers body temperature during sleep. Of course, sleep itself lowers body temperature, but does it do it enough to really activate CIRBP is the question. Is this one of the benefits of having a good night's sleep? Something that I suspect none of us on this webinar have.
Well, good night's sleep is very important. Well, we don't know a link between melatonin and CRBP. I think that's something interesting to investigate. Um, how these, well, CRBP has actually a role in circadian biology, which again, kind of completely separate function of this protein. It does control some circadian processes, which may link it to melatonin in some way. So going back to melatonin, like, is this, good for you? Uh, the levels of melatonin production go down with age quite linearly. Then, of course, not everything that goes down we need to compensate for, but melatonin shows some promise. And particularly when it is disrupted, we know it's bad. For example, in people that work, at night shifts. So there are very detrimental effects to aging, and these people have higher cancer risks. So there may be benefit in taking melatonin, although the research is still kind of not entirely clear on that.
Yeah, and I do have to point out, because it's my job to point out the weaknesses of laboratory adapted organisms, is that most laboratory mice do not synthesize melatonin. They've lost the ability during the domestication process. So, all right, so here's a really humans already live a long time, um, should we be working to make them live longer? Is that something that we should be doing? And this is more of an ethical than a scientific question, but it's one that I know I get asked a lot, and I suspect you guys do as well. How would you respond to that?
No, I think that longer lifespan per se is not that critical. What we need to think about the health span. If you are able to live healthy lifespan longer, then yeah, why not? Because if you're able to reduce or eliminate all the age-related diseases, which I think it's possible, then it will be really nice. It means we'll have no age-related diseases. We'll just live and then drop dead in the end, or we have very minor, effects of the age-related diseases. I think that's probably a dream for everyone. But again, it's important to separate between the lifespan, the health span, and I think the health span is probably more important.
Well, yeah, we really want people to be healthy and improve health. And if you think of most diseases that afflict people these days, these are diseases of aging. So why do we treat individual diseases? Let's treat the cause, which is aging. And that's really where, for me, I, derive most inspiration that, maybe we can treat all diseases at once and make people productive for longer time, which I think then it is ethical. We want to make people live their life to the fullest. And then it's not like, well, we'll have many people that are retired and need social security. Well, if people are healthy, they'll continue working. And that would solve the problem of social security. And if longer life comes as a benefit to it, then why not? I'll take it.
I would agree. So, uh, Vera and Andrei, thanks so much for joining me in this. We could go on for several more hours, I'm sure, but time is drawing near. I just wanted to remind everybody that if you joined late or you want to listen again, that the Nathan Schock Center Coordinating Center in AFAR will share a recording on all of the websites of this webinar, and we'll also put it on eBlasts and social media. And so I'd like to, again, I thank everybody. I apologize we couldn't get to all the questions. Some of them were more technical than I thought we could handle on the air, but they were all very interesting questions. And just point out that for updates on future seminars, here are a couple of websites where you can find all the information that you want. So thanks again to everybody for joining us. I hope that you'll join us on the next AFAR and Nathan Schock Center's webinar. Thanks, everyone.